I just can't see myself. All right, so welcome to the final summer 2022 colloquium. We have two very exciting talks today, one virtual and one in person. But before we hear from Sierra and Daniel, it is my pleasure to introduce the colloquium lineup for 2025, also known as our first year students. So I'm going to ask each one of them to come up here and stand at this podium and say your name and picture yourself having just delivered a stellar seminar on your research at the end of your third year. And what happens at the very end of every seminar? Acknowledgements. So take this opportunity to say thank you to just one person for something that brought you here. It can be somebody from your past, be somebody here. So I'll kick it off. My name is Nicole Perna, and I'm going to say thanks to John Poole for chairing the admissions committee that brought all these students here. And I'm glad I finished my third year.
I'm Jackie Lemaire. I'm from the University of New Hampshire. I also worked at a biotech company in Boston for the past few years. I just want to say thank you to my PI at UNH, Steve Jones, who also went here and gave me some great advice. He was excited for me to come here, and he told me to go push over some cows for him and do inebriated curling. So hopefully Madison has that in store.
Hi everyone, I'm Will. I just want to acknowledge my mentor, Katie Fisher. She started up a lab in New York, and she helped me out with learning all the ropes and all that good stuff. So, yeah.
Hi everyone, I'm Emily Ubalodi. I just want to say thank you to my PI, Bill Alexander, who was a postdoc here.
Hi everyone, I'm Katie. I want to say thank you to my parents for supporting me.
Hi everyone, my name is Alyssa, and I want to say thank you to my PI at the OPI at the University of Alabama, Ryan Early, who just gave me lots of really good advice and let me do a lot of really cool research. So,
hi, my name is Avery Imes. I studied at Miami of Ohio, and I'm gonna thank Katie, my first cohort friend. She drove me here today.
Hi, my name is Sheldon. I would like to thank my PI here, Prof. Claire Richardson, for literally bringing me here. Thank you.
Hi, I'm Dakota Nuttall. I'd like to thank my PI, Jason Hansen at Brigham Young University, who's the one who encouraged me to actually pursue a PhD and gave me the opportunity to lead my own project.
Hi everyone, I'm Rory Greenouch, and I would like to thank my parents for supporting me in science, even though they totally don't understand science at all.
Hi everyone, I'm Charles. I want to thank my PI, Professor Uyo, who I wish to see me here. Thank you.
Welcome.
So it's my pleasure to virtually introduce Sierra Love, who will be starting her fifth year in my lab. So I would say Sierra is a very courageous and unstoppable scientist, so much so that even though she is not feeling so great today, dressed up and wanted to give this presentation virtually. And so she's joining us here today. So she's been a wonderful student to have in the lab, and you'll see as use some technologies to really kind of do some exciting new work for us. And in addition to this, she's also really been the leader in a multi-PI collaboration between my group, a group at Pitt, and a group at the Institute for Cancer Research in London. And you'll see a little bit of that today as well. So with that, Sierra, take it away.
Hi, can everyone hear me okay?
I'm having technical difficulties, sorry. Let me share my screen really fast. Share PowerPoint. Let's do this. All right, is everyone able to see my screen, like my slides and not my notes?
Looks good.
Okay, perfect. Is that good? Okay. Thank you for joining me here today. Despite me getting COVID, I really appreciate everyone who's in the audience and those audience in person, and those who are here in virtual land. And thank you, Erin, for that wonderful introduction. So today I'm going to give you a brief overview on my research project, whose focus is on the discovery and optimization of novel splicing inhibitors. But before we get into the nitty-gritty of my project, I kind of want to take a step back and give you a background of the life cycle of a pre-mRNA transcript. Oops, sorry. So here we have the central dogma, where we have DNA that is transcribed into RNA, and then that RNA is then translated into proteins. But before that translation can occur, RNA has to undergo several processing steps. And this can occur immediately after transcription has begun, and even co-transcriptionally, or meaning that it happens at the same time as transcription. So we begin at the transcription start site. RNA polymerase recognizes and binds this site and region on a DNA and begins transcription. And then the first processing step is RNA capping, and this occurs at the 5' end of an mRNA. And this is important for protection of the RNA and also transport of that RNA. The next step is RNA splicing, which you remove the non-coding regions or the introns from a transcript, leaving those exons or those coding regions together. Next is cleavage, and this happens at the end of transcription. And then the final step is polyadenylation, and this is important for export of the RNA outside of the nucleus. And what we're left with is this mature mRNA that can be translated into protein. Today, the bulk of my talk is only going to focus on pre-mRNA splicing, and this is carried out by the spliceosome. The spliceosome is a large macromolecular machine. It's made up of several different RNAs and protein complexes. And these RNAs and protein complexes make up a small nuclear ribonucleoprotein, or a SNRP. And the spliceosome is made up of 5 SNRPs. You got 5 SNRPs. You have U1, U2, the U4, U5 disNRP, and the U5 SNRP. And each of these SNRPs consists of a uridine-rich snRNA, as well as a host of SM proteins that are important for stability of those RNAs. Each SNRP also consists of a host of proteins and protein complexes that are important for each SNRP to carry out its function throughout the splicing cycle. This is just a simplified version of this spliceosome. And for the bulk of my talk, I'm only going to focus on a U2 SNRP component called SF3B1, which is located in this region. Splicing is a key player in gene regulation and gene expression, and it has to be done in a very precise manner. So when splicing goes wrong, it could have detrimental consequences on the human body. And when you have mutations in splicing factors, this may result in diseases known as spliceosomopathies. And these diseases can have an impact on various tissues within the body, such as the retina, the craniofacial skeleton, spinal cord, blood, etc. And the most mutated splicing factor is SF3B1, and it is implicated in various blood diseases such as myelodysplastic syndrome, acute myeloid leukemia, or chronic lymphocytic leukemias. And as I showed you on the previous slide, the spliceosome is made up of various proteins. And when you have mutations within those proteins, it could lead to diseases within these tissues. And here are just a select few of those proteins that can be mutated and cause disease. So next, we want to know if we can target pre-mRNA splicing, or if we can target the spliceosome for potential therapeutics. Previous researchers have shown that if you have two different cell types, if you have a wild type cell, meaning there are no mutations within the splicing machinery, or if you have a spliced cell or mutations within that splicing or spliceosome, you can see that with these two different cell types and splicing occurs, it occurs relatively naturally and can excise those introns out and leave this mature mRNA transcript with each of those exons. And the cells grow perfectly fine together. But in the presence of a splicing modulator or splicing inhibitor, it could lead to various splicing patterns, meaning there could be intron retention, meaning the spliceosome splicing was blocked, leaving those introns together, or you can be left with a transcript that may lose a detrimental part of its genome or of its gene in that exon or exon skipping. And in the presence of these modulators or inhibitors, what was shown was that between the two wild type cells and those spliced 6 cells, those modulators actually selectively killed the spliced 6 cells. And some of those splicing inhibitors include those that target the SF3B1 protein that I showed you earlier. So some of those drugs include pledenylide B, herboxadiene, miamycin, spliceostatin A, and H3B8800. And as you can see, these compounds all have very different structures, but they all target SF3B1 and bind in the same region. So what our lab wanted to know is whether or not we can build tools to study pre-mRNA splicing inhibition and whether or not we can identify these novel compounds that target spliceosome machinery. So my project is based on 2 parts. The first part includes a novel inhibitor variant assay, and I'll kind of walk through the hypotheses, data, and conclusions. And this portion of the talk is done with, or this portion of my work has been done with collaborators, as Aaron has stated previously. And then the next portion of my project involves developing a high throughput screen to detect novel inhibitors of the spliceosome. So the spliceosome components are conserved across all eukaryotes, but specifically SF3B1 is very conserved between humans and yeast. And here is just showing a structural overlay between the 2 proteins. So SF3B1 is the human protein and HSH155 is the yeast protein, the yeast that we're using in Saccharomyces cerevisiae as our model. And each of these proteins consists of these alpha helical repeats or heat repeats. And what our lab has done is build a tool that allows us to study inhibition with splicing inhibition within our yeast cells. And what we've done is incorporate human heat repeats within our yeast protein and create chimeras. And here's just showing the viability of those yeast cells that contain both the wild type and the humanized version of SF3B1. And HSH155 in yeast is a protein that is essential, meaning if you knock that gene out, the cells do not grow. So we have this empty vector that does not have any of the protein HSH155 or SF3B1. So when we select for that specific protein that we use on a plasmid, we can see that there's no growth when there's no protein, but the humanized version of the protein and the wild type version of the protein grows in our yeast cells. And we're utilizing this humanized system because the wild type protein or wild type HSH155 is not sensitive to splicing inhibitors, but the humanized chimeras are. So just showing some data from a previous grad student, Sarah Hansen, she showed that if you expose these cells, the wild type yeast protein and the humanized protein to the drug pledenylide B at various concentrations, and we're looking at growth on the y-axis, and we can see that the wild type cells in black grow perfectly fine in the presence of pledenylide B, even at high concentrations, but the humanized version is inhibited in a dose-dependent manner. So utilizing our tool of this humanized system in yeast, we want to know, or our lab wanted to study splicing inhibition overall. And some of the questions that we asked is, can we modify compounds? Can modified compounds be used to target mutant proteins? And we have been working with collaborators at the University of Pittsburgh to kind of answer this question. So here I'm showing the relationship between the inhibitor and the protein, the SF3B1 protein. And that inhibitor is sequestered or bound between those heat repeats that I talked about earlier, 15 through 17. And that is how inhibition can occur. And what our collaborators have done is create different variants of drugs that target SF3B1 similar to this inhibitor here. And so we have 3 versions of this drug, miamycin D, 2'-methylmiamycin D, and 3'-methylmiamycin D. And what the difference between these drugs are the R carbons on the compound. So in R1 and R2 of the compound, we can see that our collaborators synthesize these compounds by bumping them with these methyl groups. And bumping just means adding that methyl group. And so previous research has shown that if you mutate the protein at this V1078 position, you can actually get resistance to drug inhibitors, such as herboxidine or fludarabine B. So our lab and our collaborators wanted to know is whether or not if we use a bumped compound, can we overcome inhibitor resistance caused by mutation at this V1078 position? So what we've done is utilize our humanized system and looked at growth over time in the presence of these drugs. So we want to know, do these modified compounds inhibit cell growth? And what we've done, like I said, we expose our cells to various concentrations up to 10 nanomolar of each drug, miamycin D, the 2'-methylmiamycin D, and the 3'-methylmiamycin D, and then looked at growth compared to the DMSO control. So if any of the drugs do not inhibit growth of the cells, we would see them lie across this 1.0 mark, and then if we do see inhibition, we would kind of see that fall off as as the concentration increases. So does miamycin D inhibit cell growth? As you can see in black, the wild type humanized version of the protein is being inhibited in a dose-dependent manner, as well as the mutant at V1078. Does the 2'-methylmiamycin D inhibit growth? And what we see is that no, this compound actually doesn't inhibit growth of either of our strains, meaning that bumping or including that methyl group at the 2' position actually inactivates the drug and doesn't inactivates the drug. And then finally, does the 3'-methylmiamycin D inhibit growth? And what we see is that yes, actually we inhibit growth of both of our cell types, the humanized, the wild type and the mutant. But what we see is that there is this selectivity of the drug for the mutant, meaning that the 3'-methylmiamycin D inhibits the V1078 more selectively, or it's more potent in that V1078 mutant. And this begins at concentrations a little around 500 nanomolar. So based on this data, our lab and our collaborators have come up with a few conclusions, which include that the V1078 mutant is more sensitive to the 3'-methylmiamycin D, and this method models the use of modified compounds for drug discovery, and it has a potential for tailored therapeutics for some spliceosomopathies. So now that we know our system works rigorously, we can see that we have inhibitors of splicing or inhibitors of growth particularly. We want to know if we can use this system to identify novel inhibitors of splicing. And so the question we asked is, can we use existing drug libraries in our system to detect novel inhibitors? And what I've done is come up with a screening platform that has 2 parts. A primary screen whose readout is growth inhibition. So the data kind of looks like what I showed you earlier. We're looking at whether or not these drugs would inhibit growth of our cells. And we use several drug libraries. So we have our Life Chemicals drug library, Riken Natural Product drug library, and the NCI Next drug library. And this is around 30,000 compounds that we've screened, and we're looking at whether or not these compounds can inhibit growth. And the workflow is shown here, where we have our humanized yeast system. We plate them in 384 well plates with our compounds and controls. So our positive controls include herboxidine, that known splicing inhibitor that I talked to you about earlier, and then fleamycin, which is an antimicrobial that should kill all of the cells. And then our negative control is DMSO, meaning it is a compound that should not inhibit growth of our cells. And then moving forward, we do some data analysis and look at whether or not any of these compounds show at least a 30% inhibition, and then those will move forward for further analysis. So if we have these hits, how do we know they're inhibiting splicing? That's where my secondary screen comes in, and its readout is fluorescence intensity, and we use fluorescent reporters in our V strings to determine that gives us a direct readout to splicing inhibition. And here is the workflow shown here. So we grow our cells overnight, dilute them down, plate them in 96 well plates, and then allow a plate reader to read absorbance and fluorescence intensity for around 5 hours, and then do some data analysis. For simplicity and time, I'm only going to talk to you guys about some data from my primary screen, and particularly that from the Life Chemicals drug library. But before I go into my data, I kind of want to take a step back and talk about rigor and reproducibility and high throughput screening. So for my experimental design, I am doing 2 biological replicates, meaning I'm going to screen all of the drugs twice and all of my strains twice to make sure if I'm seeing any growth inhibition, it needs to show up in both of those replicates. And then I am ensuring that I'm having consistent growth conditions, meaning I am trying to avoid batch effects and making sure that I'm using the same media for all replicates and for all growth conditions. And then finally, to avoid human error, I utilize automated cell and liquid handling, and this just means that I have machines that will put cells onto my plates as well as those drugs, just to avoid missing a well or things like, or missing a well. And then finally, with data analysis, we store our data into the collaborative drug discovery vault. And what this does is kind of clean up our data, normalize the data, and give us a quality control output to see whether or not our assay is rigorous and if it's reproducible. And then I also utilize our statistical programming for visualization and statistical analysis. So the first step of high throughput screening is ensuring that our assay is of high quality. And what we do is calculate a Z-prime score calculate a Z-prime score. And what that does is actually look at all of our controls, negative and positive, and to ensure that both of those controls are very well separated, meaning we see growth in those wells where we have DMSO, and we see inhibition in those wells where we have fleamycin or herboxadiene. And that kind of looks at the entire plate as a whole and gives us this Z-prime score utilizing this calculation. And any plate that has a Z-prime score of around 0.5 or higher indicates that our assay is rigorous, and that the plates themselves do not have any issues as far as growth across the entire plate. So after screening over 100 plates, we can see the Z-prime score for each plate located here. And as you can see, each plate Z-prime score is well above this 0.5 marker or 0.5 area, and well over 0.75. And this just indicates that my assay is of high quality, and then I can move forward with data analysis. So moving forward, we want to know whether or not any of the compounds in the LC4 library actually inhibit cell growth. And first, we want to look at whether or not they inhibit cell growth. So we're looking at 25,000 compounds, and then looking at percent inhibition. And this just means if the cells grew, they would be at 0% inhibition, and if the cells did not grow, they would be anywhere in between. And my threshold for identifying hits is around 30% inhibition. So for our controls, we would expect to see DMSO wells showing up in this 0% inhibition. And then for fleamycin, we would expect to see it around 100% inhibition, as well as that herboxadiene. And looking at the data from the 25 compounds that was screened, we can see that majority of those compounds lie within this 0% inhibition, similar to that of the DMSO control. And we have a few compounds that inhibit growth that varies between around 40 and 100%, and those will move forward with secondary screening. So overall, do any of these compounds inhibit cell growth? We have around 61 compounds that show at least 30% inhibition. Out of the 25,000, 61 is a really good number, so our hit rate is less than 1%. And then moving forward, these hits will be screened in our secondary screen and to identify whether or not they inhibit splicing directly. So to summarize this portion of my talk, I've successfully screened multiple drug libraries, but specifically I've talked to you about the life chemicals library. We have around 61 hits. And then moving forward with all of the compounds from the preliminary data, around 150 compounds will move forward with secondary screening to ensure that to see if they inhibit splicing. And with that, I would like to thank my lab for all of their help with my project and any feedback they have for me. I would like to thank my collaborators at the UW Carbone Cancer Center or the screening facility with their help, as well as my collaborators at the University of Pittsburgh, Dr. Kaz Queta and Jacob Beer for their synthesis of the neomycin variants that we've used. I would like to thank my committee members and my funding sources. And with that, do I have any questions? I can't hear anything if someone's asking questions. Is it muted? Okay.
Nicole, you're muted.
Why can you explain why these chimeras as opposed to the full protein would not just not work with the full protein, or like the precise purpose of chimeras?
Yeah, the question is why a chimera instead of using the full protein?
That's a good question. So we utilize the chimeras in our yeast system only because if we use the full protein, it doesn't express well. And it doesn't express well or express at all, so we don't get any yeast growth. But we also use these chimeric proteins instead of the full protein in our yeast system because it's nearly impossible to do in human cell lines. If you make any type of mutant to the SF3B1 protein in human cells, it is detrimental. They may grow, but they might be super sick, and it's very unlikely that will allow us for screening or for testing, for screening at these levels.
Thank you. We have another question here.
Make you want to go forward with the drug and say this is probably therapeutically relevant.
What level of splicing inhibition do you want to see in order to advance a candidate that might be therapeutically relevant?
So we actually want to see that selectivity that I kind of showed you a little bit earlier. So if we're inhibiting splicing, splicing occurs throughout all of the cells within our body. So if we can selectively inhibit those that are mutant, that would be the purpose of the therapeutics that we are looking for. And does that answer the question?
Like, is there a percentage threshold?
Is there a percentage threshold of inhibition?
Not quite sure, but I'm assuming, not quite sure, but I will look into that.
All right, I think we'd better move on. So let's thank Sierra for a fantastic talk.
Thank you, everyone.
Okay, go ahead. So today, the next speaker is the Daniel Osorio Mendez, the fifth. Daniel is a really great and talented graduate student, and then his fifth year just started. Maybe we started this September. Okay? So for last 4 years, he did a really good job, and then the very challenging project that I believe in my lab, the most challenging project, because he tried to identify the novel regeneration gene using the 4-gen screening zebrafish. So it's really difficult to isolate the mutant, but more difficult thing is the dissecting the raw molecular mechanism. So Daniel tried the multiple different way. Then ultimately published the really great paper this year, a couple of months ago, in which he demonstrate how nerve affect the appendage regeneration, particularly in the fin regeneration. So he's gonna talk a little more about that story, and then also he started a new project, a new research direction, which is really intriguing for everyone in biology, particularly in the regen medicine. So let's hear the exciting story from Daniel. Okay.
Okay, thank you, Jinsu. That's a huge pointer.
The resolution on the screen there is
Daniel,
we're seeing your presenter view.
Oh,
give me 1 sec.
cool.
That should be good now.
Good. Good. I'll just gonna use this. Hi, my name is Daniel Osorio. And my project involves it's kind of a broad title, the role of nerves in controlling zebrafish fin regeneration. Can everyone hear me? Okay. So as we know, regeneration is not very well conserved among species. Some species can regenerate multiple tissues such as appendages, brain, hearts, and those include zebrafish, some and other fish species, and salamanders. And then mammals, such as humans, we like this ability to regenerate a lot of organs. In our lab, we study the zebrafish fin because it serves as a model for appendage regeneration. And because it regenerates pretty quickly within a span of about 3 weeks, it is a pretty simple model to use. So this is a caudal fin or the tail fin of the fish, and it has multiple fin rays, which is the bone in the fin. And so during regeneration, during appendage regeneration, you have you need a very important component that's called the blastema. So this is a section of the regenerating limb in salamanders. This is the bone. And you can see this mass of cells. So that's the blastema. It's a heterogeneous mass of cells that are going to divide and give rise to the new tissue. And that's, if you don't have a blastema, you pretty much don't have regeneration. And another important compartment in this process is called the wound epidermis. So that's kind of a thick layer of skin on top of the blastema, and that's also a very important signaling center. It's not just skin, it's an actual component of regeneration. And another crucial aspect in regeneration of limbs in salamander and zebrafish and almost all species is the presence of nerves at the site of injury. So this is again a salamander limb. So when you amputate, you have limbs shown in yellow, and they kind of invade the growing tissue, and they are essential for this process of regeneration. And this has been shown also by doing some denervation studies. Again, this is a salamander limb. In red, you have the nerve going into the limb, and then if you amputate in the normal limb, you have regeneration as you would expect. However, if you denervate the limb before amputating, so that the nerve degenerates, as you can see by these red dots, then you pretty much don't have regeneration. And this is called the nerve dependence of regeneration, and it has been known for a long time, since the 1820s, where a British scientist by the name of Tweedy J. Todd, he started experimenting with limbs and nerves in salamanders. And another way in which people prove how crucial nerves are for regeneration and for blastema formation is by doing an accessory limb model, and that is where you make a wound in the limb, just a simple wound, no amputation, just a skin wound, and then you deviate the nerve to go to that wound site, and then that forms an ectopic blastema, so a blastema where you shouldn't have one. And that again proves how important nerves are for initiating regeneration. And another thing regarding nerve supply in the limbs is called the quantitative nerve threshold. So that's where people observed that you need a certain amount of nerve endings at the site of injury to induce a regenerative response. So in the innervated appendage, that leads to regeneration upon amputation. However, if you denervate or partially denervate the limb, and you have, let's say, some nerve endings, but not quite above whatever the threshold is needed, then you still fail to regenerate. And what controls this innervation into the blastema is not well understood. So I'm going to go back a little bit and talk about my project regarding the 4 genetics screen. So in our lab, one of the things we try to do is find novel regulators of fin regeneration. And to do that, we employ 4 genetics. So that's where you focus on the phenotype, and then you look for what gene is causing those phenotypes. In our case, our phenotype is any defective regeneration. And so we did one of these screens, and we found a mutant that shows fin regeneration defects. But interestingly, this mutant also showed a swimming defect. As you can see, this is a control mutant. And both of these were temperature dependent. Only at temperatures above 30 degrees were you able to see these phenotypes. And then both of these appear normal during 26 degrees, which is the water temperature that people normally use in fish. And so this is a 70 PA fin, or 7 days post amputation. This is control mutant. This red triangle is marking where the fin was amputated. So you can see the mutant shows a pretty obvious defect. We try to name this mutant, so because the phenotype, the swimming phenotype looks kind of similar to what cataplexy looks like in humans, which is a sudden and transient episode of muscle weakness or paralysis. We decided to name this mutant temperature-triggered cataplexy, or TEMCA. So I'm going to be referring to this mutant as TEMCA for the remainder of my talk. And then obviously the next question is, looking for what gene is causing these phenotypes. And through mapping and other techniques, we found that a mutation is present in a gene called SCNAB, and that encodes the NaV 1.6 voltage-gated sodium channel alpha subunit. So this is a structure, it's a pore, a channel that is involved in the transport of sodium through the membrane. And as you might expect, this is a neuronal gene in mammals, where it is expressed mostly in neurons, especially at this region close to the body of a neuron called the axon initial segment, as well as at the nodes of reindeer. And so this is where the axon potentials initiate, and then the nodes of reindeer are where those action potentials are propagated through the axon. And so during my previous project, I was trying to characterize this mutant. And briefly, we found that one of the causes of the observed defective regeneration was, or is likely due to the decreased innervation of the regenerate fin. So this is one fin ray, and this is showing the where the images were taken in the fin, and then this is the amputation plane. And we saw that when you label nerves using acetylated tubulin in the 2DPA fin, you see the controls. They have a pretty thick bundle, which is what is inside of the fin rays, and then a lot of neural regrowth. However, the TEMCA mutants, they show pretty remarkable decrease in innervation, and that's likely at least contributing to the decreased fin growth of these mutants. And so we found that these fin defects are driven by decreased innervation, which in turn leads to decreased plasma proliferation, which we also showed. But for my next project, I want to take a different route regarding this mutant and this gene, and that is looking into the expression pattern of SCNAB during both in regeneration as well as development. And that is because expression data for this gene is lacking in zebrafish. There's only a few, or I can only find one paper showing any sort of expression of SCNAB. This is from my project. We collected the adult brain to see if our gene is also neuronal in zebrafish, which it is. So you can see the SNAP transcript in purple in the brain pretty clearly. And then one of the only papers that I could find showing any SNAP expression pattern is from 2006, and they show that SNAP also shows neuronal expression. So you can see it in the trigeminal ganglion next to the eye, as well as the spinal cord, in these RB or Rohambeer cells, which are also cells of the nervous system. So in order to look at the expression pattern of SNAP more closely, we wanted to generate a reporter line. And for that, we actually tried using several techniques, such as making a transgenic line or using CRISPR-Cas9. And we for those lines, we didn't see any expression, which we assumed it was due to the gene being very lowly expressed. So then we found this paper by Kimura et al. So where they used an HSP73 promoter to upstream of a reporter gene such as GFP to improve the expression of that reporter in your cell types of interest. So we attempted to use the same approach. So then we used CRISPR-Cas9 in this donor DNA having our GFP reporter gene, as well as the HSP70 promoter upstream of GFP. And doing this, we were able to see very nice expression of our gene. So this is the 5-DPA for 5 days post fertilization larvae. And you can see GFP in the nervous system, as you would expect. So this is a spinal cord. You see again the same Rohan-Beer cells that you see in this previous paper, as well as the trigeminal ganglion next to the eye. So we're very excited by these results. But we studied the adults and the adult fin specifically. So we wanted to see if these GFP expression persists or is maintained throughout adulthood. So I looked at the adult fin, and we also see very nice expression in the nerves. These are the thick nerve bundles in the anterior fin inside of the bony fin rays. And then the 3GPA fin, you see again the same bundles, and then the axons or neurons growing into the newly generated fin tissue. So now using this line, we want to answer several questions regarding kind of the role of nerves and how they contribute to regeneration, which are questions that, to our knowledge, haven't been answered in our field. And some of these are, what are the target cells of SNP positive neurons? Are they blastema cells? Are they epidermis or wound epidermis or something else? And also whether SNP positive neurons are mostly comprised of motor neurons or sensory neurons. And this is, again, due to the lack of data regarding which neuronal subtypes are more important for triggering a regeneration response. And then we also want to see whether SNP positive nerves directly induce regeneration by secreting factors or any other molecules, or whether this interaction is mediated by Schwann cells, meaning that the neurons might be activating or communicating with Schwann cells, and then the Schwann cells themselves are the ones secreting those pro-regenerative factors into the blastema. So again, we can look into the interactions. So the nerves are in red, and this is again the blastema in the regenerating limb. So there's no data to our knowledge of whether the nerves are communicating with the blastema cells or with the wound epidermis. And also another possibility is that, again, the nerves are communicating with Schwann cells, and then those Schwann cells are secreting or interacting with either one of these or any other cell types in the growing tissue. Or both are possible as well. And for this, we are I'm going to be using our lines in the heterozygote and mutant backgrounds to also see how the TANCA mutation affects this innervation and interaction between nerves and cells in the growing fin. So I will be amputating and then looking at the regenerating fin at different time points throughout regeneration. And then same for markers for blastema cells, the wound epidermis, as well as motor sensory neurons and possibly other cell types. And then in my previous project, we found that the lack of innervation is probably one of the main causes of the lack of regeneration in these mutants, but we still don't have a clear idea of what's happening molecularly or in the transcriptome of those nerves that's causing them, or that's preventing them from growing into the newly generated tissue. And so in the field, this is also related to the nerve dependence model of regeneration, where you have it's called the neurotrophic model of regeneration. So, and this is where you have again, neurons secreting regenerative factors into the regenerating tissue. And some of these factors have been known to, or are thought to be, or to include NRG1 or neuroregulating 1, or some bone morphogenic proteins, some fibroblast growth factors. But the one thing to notice is that while these factors have been shown to be secreted by neurons, they are also secreted by other cell types in the blastema or in the wound epidermis. So they're not unique to nerves. So to look more into the any transcriptomic changes going on in the nerves of the fin in our mutants. I plan to do trap-seq or translating ribosome affinity purification sequencing. And that is, so the way this works is you have pretty much you express a transgenic large ribosomal subunit gene, so that's L10A, that is used to GFP, so that you can use anti-GFP to pull down GFP and also those ribosomes as well as the mRNA is being translated by those ribosomes in a cell-specific way. So, for example, you have a heterogeneous tissue with different cell types, and only your cells of interest are expressed in GFP and also these ribosomal subunits. So you can use this method to only pull down the transcriptome from your cells of interest. So you don't have to worry about any bulk effects from other cell types. So we wanted to take this approach as opposed to single cell RNA-seq, for example, because in the adult fin you have, as I showed, axonal projections in the fin, inside of the fin race. However, the bodies of the neurons, those are in the spinal cord, and only the axons kind of invade the caudal fin, so there are no neuron somas in the fin. So if you try to do single cell RNA-seq, for example, you wouldn't get anything because there are no cell bodies in the fin, only the axons. But using our approach, we can collect the fin tissue and then just get that RNA from the nerves. And so if only we had a line showing expression of nerves in the fin. So we do. So that's the one we just made. So we just added the L10A gene, again, the ribosomal subunit, at the end of GFP, so that we can use this construct to do trap-seq. And I don't have a lot of data yet, but I do have the F0 fish, and they still show the expected nervous system expression in the spinal cord. So this is 5-DPF larvae having our construct. So that's good so far. So once I generate stable lines, I want to use this fish to collect tissues from Tenka and control fish, and as well as uninjured fins and 2DPA fins, and possibly other time points as well if needed. And then collect the mRNA bound to those GFP-type ribosomes, sequence, analyze, and hopefully try to identify any differentially secreted factors between our Tenka mutants and our controls, and see if those are important for regeneration and if those are the ones causing those regenerated phenotypes. And another thing, now this is regarding the developmental effect of SNAB in zebrafish. So when I was doing experiments, I noticed that if I raised SNAB mutants 33 since the larval stages, the very early stages, I noticed this kind of development phenotype. So this is the control adult, this is the Tenka mutant. In the adult, you have very clear or very nice pin race, but then in the mutants, there seems to be some bone fusion going on at the tip, or something going on with the bone. It looks nothing like the wild type fish. So I would also like to use our fish lines to see whether nerve patterning is affected in this fish during development, and also use this in addition to some osteoblast reporters that we have to show bone cells and how they are, or whether they are affected by the innervation pattern of this fish, as well as, and we can also stain for bone deposition using this Alisson red stain, so that since the mineralized or calcified matrix. And we think this is an interesting question given a recent paper published in Development a few months ago, where they show that osteoblasts control nerve patterning, but not the other way around. But we are seeing, possibly nerve function affecting osteoblast or bone deposition. So we want to we're trying to see what's going on, or if we can find any cool data from this. So now moving on, I want to go into rigor and reproducibility regarding my project. So in our lab, we do a lot of fin amputations. So one of the things I've tried to be careful with is, being consistent with my amputations in terms of how much I amputate, as well as how clean and how horizontal the amputation is. And this is because in zebrafish, the more you amputate, the faster the fin is going to regenerate. So if you're comparing two groups and you're not very careful and you're amputating, like kind of half for some, maybe a little bit more for other fish, or a little less for other fish, or if you do this, this side, since you amputated less, is going to regenerate more slowly than this one, because they both need to regenerate fully in a span of 3 weeks, roughly. So of course, this side is going to need to grow faster to reach the same time point as the other side. Just trying to be consistent regarding these amputations. And if you need to, you can see how consistent your technique is by comparing whatever is left of the fin after amputating relative to the original uninjured fin size. So I did that once for my fish. So this is the 0 dpa fin, so the fin length right after amputation over the original fin size. So if I'm amputating half, it should be pretty close to 0.5, which it is maybe a little bit less, but that's okay as long as you're consistent. And you can also see that between fish in the same groups, there's not a lot of variation, which is again good. And so you might have to do a little bit more work, because then we don't usually image the uninjured fin, but if you need to do this analysis, it's like an extra step to do, but it's good to do, so you can make sure that your technique is good enough and you are more confident of your results. And also, I try to keep track of the gender of my fish. So in case you have control fish and mutants, and then you're measuring regeneration, and you're seeing something like this, and you think there's nothing going on. However, maybe if you label by sex, and you see something like this happening, then there's something else going on, and you can look more closely into that. So yeah, again, looking or keeping track of gender. And what I also like to do is keeping track of the orientation of the fin, or what side of the fin I'm measuring, such as the dorsal side or the ventral side. So when I'm imaging the fin, I always image so that the dorsal side is to my left on every single image, and the ventral side is to my right. And then again, in case there's something going on, such as the dorsal side or the ventral side having a more pronounced effect regarding regeneration. So in summary, some summary and future directions. So we identified SNAB as a novel neuronal regulator controlling fin regeneration. And we generated a new zebrafish SNAB reporter line that we plan to use to look more closely into the nerve dependence of regeneration, and see where are the target cells of nerves and how those cells respond to whatever signals are being secreted by nerves, as well as discovering more of those signals, and also determine the developmental role of SNAB in the zebrafish fin. And that's it. So I would like to thank everyone, of course, in the Kang lab, especially Ryan, who helped me generate those reporter lines. My thesis committee, collaborators from Duke and other places, and also the Hashim Lakar lab for sharing the LTNA plasmid construct that we plan to use for TRAPSIC. Thank you, and fun.
have a question to that. I have
Yes.
a small question.
Mhm.
First question is like, why were the fish in 33 degrees in the first place? Why did you put them at 30?
Yeah, so I didn't explain that. So during the initial 4 genetics screen, because a lot of genes involved in regeneration are likely to also be involved in development. So if you mutate those genes and they're lethal, then you will never recover any mutants. Even if they do control regeneration, you won't see them because the fish die before you can actually test for that. So what people do to sort of broaden the pool of genes that you can isolate from those screens is you do kind of a temperature shift. So you amputate the fin and then immediately you place the fish at 30 degrees. So you're looking for, heat sensitive mutations in our case. And then you can kind of mess with the activity of the gene without killing the animal, and you can recover more genes potentially that are involved in regeneration, which is true for our gene. So we have deletion. So our Tinka mutation is a point mutant, but we also have deletion mutants, and those are fully null. And we see that if you have homozygote mutant fish, they die at about like 1 month after being born, which again, if that happened, you wouldn't be able to find our gene as being important for regeneration. Mhm.
Thank you.
Sure,
My
thanks.
question is, your nerve pictures were so cool with like the fluorescent
Oh,
nerve.
thanks.
And I wanted to know what type of microscope did you use?
I think those are just using. I don't remember.
It was the fluorescent ones.
Oh, this. These I took with a confocal, and then these I took with a normal AP fluorescent microscope. So I can probably get even better than these two if I use a confocal and like maybe like stain for GFP to like decrease background or something.
Very cool talk. Thank you.
Thanks. Yes.
Actually have a two-part question.
Of what?
Cartilage.
I think I don't know if they do in the fin. I'm not sure about any other body regions, but I think they do. Yes. Mhm.
So, even feedback model.
I have not. Usually, do you know if there's cartilage in the fin bone?
And the generation between the mammalian bone and then used the appendage region including salamander and then fins are totally different. The mammalian, they don't have a button. So they just heal, right? Even the two just like the heal bone, but actually they cannot hold extra whole pieces. So, but anyway, they have the mechanism to repair the bone.
And also the bone in the fin is not the same as I forgot the name, like the actual bone, like inside of our arms, for example, is dermal bone, and it has a different process of being made compared to like actual muscle skeletal bone. Mhm.
Okay, well, let's say thank you, Daniel.
Thank you.